All right, everyone, welcome to today's lecture, which is a technically optional lecture on the brain, although I hope that most of you end up watching this because hopefully you'll learn something. For those of you who have some background in neuroscience or have taken cognitive neuroscience or behavioral biology, some of this should be a review for you. For those of you who maybe aren't as familiar with the brain, this is just intended to be a broad overview of a little bit of the history of neuroscience, the parts of the brain, especially the parts of the brain that matter when we study cognition. And the goal is not for you to learn every single little component of the brain. That's not necessarily important in cognitive science and cognitive psychology, but what matters is that you have a basic familiarity with the brain, with how it works, and some of the key areas and structures that we might reference because they directly relate to cognitive processes. And we will be reading some neuroscience papers, reading some cognitive neuroscience papers, and referencing the brain and how different neural processes give rise to cognitive function in different sections that we'll talk about in this class. So what we'll cover in this lecture is, like I said, a little bit of the history of neuroscience, the concept of emergence and how it relates to cognition, the levels of analysis in the brain, so different ways that we look at the brain, the parts of the brain, and then some key techniques and technologies that are used in recording and neuroimaging of the brain. So just a little bit of history. So neuroscience is sort of as a science, maybe about 200 years old formally, although since ancient times, people were interested in looking at the brains of people who had passed away, for example. And most of what sort of constituted early neuroscience was anatomy. So all you could really do, you can only access the brain in people who basically were already dead or in the nervous systems of animals, dead or alive. So from just opening up people's skulls and looking at their brains, there was some sort of general mapping of the brain, of the different structures, and people hypothesized about what those structures could correspond to. The brain looks really weird and interesting. And the sort of pseudoscientific study of phrenology sort of came out of this notion, which is an accurate assumption that different parts of the brain do different things. And although they got it all wrong in terms of trying to map different functions onto different parts of the brain and sort of therefore the head, they had the right idea, right? And phrenology is a sort of extension of that, which is to say, when people have bumps on their skull, and bumps on your skull are from, well, this part of the brain is sort of bigger, it's protruding more. So if you sort of have, a more protruding back of the brain, that means that that part of the brain is bigger, and you must be really good at whatever that part of the brain does. So they were trying to make reverse inferences into, well, this person is really good at math and they have this bump on their skull here, so that must be the math part of the brain. The other ways that we were able to make early inferences about neural function was from lesion studies. And lesion refers to when a part of the brain is removed either from an accident, from some sort of tumor, from a stroke, somehow sort of a part of the brain disintegrates or is missing. And when a part of the brain is gone, and especially in an adult, there's a sort of a before and an after. This person was able to do something before, and now they're not able to do something. Therefore, the part of the brain that's missing must be responsible for that function. So a very early and influential example of this is the famous case of Phineas Gage, who had a railroad spike went through his skull under his eye socket, and it lesioned a part of his frontal lobe, and he survived miraculously, but his personality completely changed. He was erratic, he was irrational, he was impulsive, he was rude, he lost sort of his social graces and his decision-making abilities, and those we understand to be a big part of what the frontal lobe does. Other famous examples are Broca's area and Wernicke's area. Both Broca and Wernicke were like medical doctors and anatomists, and they have these regions of the brain named after them because they respectively put together that patients who had these particular or parts of these parts of their brain missing due to stroke, for example, had consistent patterns of cognitive dysfunction or disability after the fact. And so you can only know that after the patient had died, but retroactively, if you see enough cases that are the same and after they die, you open up their skull and they all have this one part of the brain missing, you can infer that that part of the brain is related to that cognitive function. And in Broca and Wernicke's area, these were related to language production and language comprehension. Slightly more advanced methods sort of arose in the early 1900s, where in non-human animals, so rodents, animal models such as in sea slugs and non-human primates, researchers were able to use sort of more invasive methods, studying cognitive function, with the animal still alive. So you wouldn't necessarily do this to a human unless they were in really bad shape, but you ethically, or, at the time, and still today, people study the neural activity of alive animals by implanting electrodes in different parts of their brain, having them do things, and then sort of experimentally making inferences about what different parts of the brain are doing. So you can study the nervous system as it actually functions in real time in non-human animals this way, but it's very invasive. You can do that in human patients as well in cases where individuals are getting brain surgery and their skull is already open. There's no feeling in the brain. The brain is made of nerve cells, of neurons, but it doesn't have sensory neurons. Like you can't feel the brain, like if someone was touching your brain. So people who are under localized anesthesia and are awake, you can plant electrodes in their brain and you can record their neural activity, the activity of sort of large areas or individual neurons, and have them talk to you, have them do things. This is something that's really common in brain surgery patients when they have to get part of their brain removed, and you want to make sure that you're not cutting out more than what's necessary clinically or to help with whatever problem they're having. So you can sort of map onto the area and sort of make sure that, based on what their behavior is, and make sure that you're not taking out too much. And that's most common in epilepsy patients even today. Starting in the mid 20th century, so the mid 1900s into the late 20th century is when we start actually seeing techniques for neuroimaging and neural recording in both sort of clinical and non-clinical human subjects. So just being able to non-invasively study the neural activity of a human and to make inferences about cognitive processes based on that. EEG as a technique was developed really early, as early as the 1930s, but it was pretty primitive and could basically be used as a diagnostic technique for like epilepsy. But it has become a lot more advanced. So sort of you see this this boy here with only a few electrodes on his head. And now today there are really dense EEG mesh caps where you're recording from like hundreds of little points and you're getting this really strong picture of the electrical activity of the brain. Imaging techniques include PET scans, MRI, and fMRI. So PET uses radioactive material that can be picked up metabolically. MRI is a static image of the brain, and fMRI is a moving image of the brain based on metabolic activity. And fMRI wasn't really developed until the late 80s, early 90s, and it was only able to be used as a neuroimaging technique at that point. And then stimulation techniques such as transcranial magnetic stimulation and transcranial direct current stimulation, TMS and TDCS, weren't developed until the 2000s. So it is at this point where we're able to actually do experiments, bring people into laboratories, people who are healthy, not people who have epilepsy or who are already dead. And we can study neural activity because we are able to record it and then associate it with their behavior or their whatever cognitive process that they're doing at the time. And that's when cognitive neuroscience really ends up being really viable as a field. And so much of what we know about the relationship between the mind and brain, at least cognitively, has happened within the last 30 or 40 years or so, and even more recently. Okay, so the brain is incredibly complex, and there's lots of different ways that we can study it at multiple different levels of analysis. And so to start, I want to just sort of start with a metaphor of what this actually looks like. So let's think of the brain as a city. So a city is this really complex ecosystem with hundreds, thousands, millions of people and systems within it. So the brain is very complex. So let's say that the brain is like a city. In this metaphor, tall buildings and neighborhoods are functional regions of the brain. So in the physical space, there's concentrations of activity that correspond to one particular function, or sort of areas that have their own character and are sort of separate from other spaces. So it's buildings, it's large buildings, and its neighborhoods are like regions of the brain. Its roads and its subways are like pathways. So in the brain, information can be transmitted like from one area to another. And in a city, that's kind of like the way that roads and subways work. There are local pathways, so you can just go down the street, or you can take, the subway from one side of the city all the way to another. And as part of this metaphor, just like in real cities, some areas are better connected than others. Some areas, there's lots of different ways to get from to places, and the roads are very big and fast. Others are sort of more disconnected from one another. You would have to go through lots of different stops to go from point A to point B. And that affects the flow of traffic or the flow of information. And that's the same as in the brain. And in this metaphor, people are neurons. So neurons are sort of the units of the brain, and they sort of flow throughout the city. They flow throughout the city depending on sort of their goals, and they're sort of the information, they carry information, right? So people carry information. And there's different types of people, right? People have different content to share and transmit with one another depending on the circumstances, depending on their goals and what they specialize in. So just like people, there are different types of neurons that have different functions, and those sort of people are going to interact with one another in different ways depending on their function. Neurons don't sort of interact with each other necessarily in that way, but the information that they carry does. And just as a little aside, I want to talk about the concept of emergence. So what is emergence? And this is something that we'll say a lot, we'll talk a lot about in this context. So an emergent property is a property that complex systems have that its constituent parts do not have. So it's a property that the system has that the parts of the system individually do not have. This property emerges as the result of behavior of components at a more fine-grained level of analysis. So, for example, we would say that The behavior of organs, organ systems, and organisms are the emergent property of components at a finer level of analysis in biology. So molecules, cells, tissue, etc. So the behavior of a cell is the emergent property of the behavior of molecules. Cells and molecules aren't sort of analogous, they're not identical to each other. The things that are happening at this level feed up to the things that happen at this level. And you can study one in order to understand the other, but the behavior at this level is the result of what's going on over here. And sort of you can go up the chain of complexity. The behavior of tissues, of organs, is the emergent property of biochemical processes at finer level levels of analysis, and all the way up to organisms. So the behavior of a human is the result of the behavior of atoms, but there's lots of different levels of analysis in between. Same thing for like ecosystems and biospheres, right? Ecosystems are the emergent property of populations of organisms that are living and interacting with one another within some environment. So the ecosystem is the result of all the behavior of the individual organisms, plants, animals, etc. But understanding sort of the behavior of plants, animals, and etc. in isolation doesn't on its own describe or explain the patterns that are seen at the level of an ecosystem. And we bring this up because cognition is the emergent property of the brain. Cognition emerges from the behavior of the biochemical processes of the brain as an organ. The brain is a tissue, it's an organ, and the sort of networks and subcomponents within the brain help us understand what cognition is. They give rise to cognition, but it's not the same as understanding what cognition is. Cognitive processes operate at a different level of analysis than the biochemical processes which underlie them. So to continue with our metaphor, if the brain is the physical infrastructure of a city, the people within it and their information content, then cognition is the economic output of businesses, of business sectors, or of the entire city. So sort of what the city produces. The city produces goods and services. It produces money. It produces things. It produces food. It produces, patterns of behavior. Groups of people living together produce culture. They produce trends and cultural artifacts, right? So there's sort of these things that emerge as the result of the sort of individual components of a city, the people, and the way that information can flow within that ecosystem. But when we study cognition, what we're studying are sort of this level of process. And in order to help us understand how these things happen, we can look to things like traffic flows, business goals, population density, things like that. So kind of similar or analogous to sort of how a city works, there's this bunch of different levels of analysis of the brain, and different parts of neuroscience and cognitive neuroscience sort of make use of analyzing the brain at different levels. So we could try to understand the function of the brain at the chemical or molecular level, at the level of neurotransmitters. We can try to understand at the cellular level, sort of the building blocks, the units of which the organ of the brain is made up of. We can look at like a bigger level. We can look at brain structures and regions and sort of how they correspond physically to different parts of the brain. We can understand the brain through its pathways, right? The way that there's sort of large scale pathways between different brain regions and what that says about the information flow between them. And we could also look at the activity of large-scale brain networks. So different parts of the brain are sort of associated with one another. They talk to each other. They share information, and the ways in which those information patterns show up is not exactly the same as just looking at the structure of the brain. So different subcomponents of the brain are more or less connected to one another because they relate to different function. So there's sort of this symphony happening. It's not just like one area and another area. There's sort of complexity in those interactions. So does studying these biochemical components tell us what cognition is at any of these levels of analysis? No, not individually. Studying any of this does not tell us what cognition is, but if we attempt to sort of understand all of it, then it paints a picture. And the level of analysis that you focus on depends on your goals and your measurement technique. So what are you trying to understand? What are your goals? Are you a chemist? Are you an anatomist? Are you a medical doctor? Are you a neurologist? Are you a psychiatrist? Are you a psychologist? All of these things will, depending on what your goals are, you will prioritize sort of understanding the brain at a certain level of detail. Understanding sort of the cellular structure of the brain is helpful a little bit, but it doesn't tell us that much about behavior, right? If you want to understand behavior, you might be more interested in pathways or large-scale brain networks, for example. So let's just break it down briefly. So the neuron is the information transmitter and encoder of the brain. Neurons are nerve cells. There's also different kinds of neurons in the brain that have different shapes and different functions, but the general sort of layout of any kind of neuron has these particular components. So all neurons have a sort of cell body, which is called the soma, the nucleus within the cell body. The dendrites are sort of the branches that pick up information from other cells. So it's kind of like the roots of a neuron branching in one direction, and then the axon branching in other directions. So the axons are what send information out, and the dendrites are what are picking information up from other neurons. Myelin is sort of this fatty substance that coats the axon, and similar to the insulation on electrical wires, myelin protects the cell. It makes the information transmit faster because it's insulated, and it protects it from interference. And then the axon terminal is sort of this very end of the axon where neurotransmitters are released and sent to other neurons. And so when neurons fire, it looks something like this. These are just neurons strung together, right? There's some sort of information is picked up from the dendrites here. It causes a reaction which causes the signal to continue and be sent, transmitted through the axon to the axon terminals, where it is the signal then passes to other neurons, and so on and so forth. So this is sort of the building blocks of how information is transmitted through neurons. If we zoom in to sort of the space between neurons and what's happening at the synapses, which is sort of that space in between neurons, what we see is that neurotransmitters are released from one neuron and sort of released into this the space between, and then picked up by receptors on the other side in a different neuron. And so that's basically the way that cells communicate with one another. And you saw that GIF there that I think shows it really well. So neurotransmitters are sort of little information packets. And importantly, there's kind of different neurotransmitters and within the brain and within the nervous system as a whole, and different neurotransmitters that have different molecular structures. They are sent by and picked up by different kinds of transmitters and receptors, and they correspond to and produce different neurochemical functions and give rise to different cognitive and behavioral effects as a result. So this is sort of the neurochemical or the biochemical level of the brain is sort of like, what is the kind of information that's being sent? What is the chemical signature of those neurotransmitters affects the kind of neural activity, and different neurons in different parts of the brain sort of trade in different, these are sort of different currencies. Just some key neurotransmitters that are relevant to cognition might include adrenaline or norepinephrine, which produces fight or flight response. It produces arousal, so like a stress response, and it facilitates an increase in heart rate and blood flow. So if you are doing some sort of stressful physical activity, and heightened awareness. Noradrenaline and norepinephrine is associated with concentration, so it facilitates concentration, it affects attention, it contracts blood vessels and increases blood flow. Dopamine is sort of known as the pleasure molecule, but it's really associated with reward and motivation. So it's associated with feelings of pleasure, learning, and when learning goes wrong, and when we sort of feel too much pleasure from something, this can lead to addiction, and it also helps initiate movement and motivation. And we learn because we sort of repeat behaviors that lead to dopamine release. That's sort of the basic way that reward-based learning occurs. Serotonin is associated with mood. It contributes to well-being and happiness, and it sort of helps regulate our sleep and digestive systems, and it's affected by exercise. GABA and glutamate are associated with focus, attention, and memory. Acetylcholine is associated with learning, thought, and memory, and then endorphins are sort of euphoria and pleasure. We probably won't talk about neurotransmitters that much in this class, but these are all really important to be aware of. So then bringing it up to sort of one level of analysis further, when many neurons fire at once in a given area, so it's never sort of, or it's very rarely sort of single neurons that are being recorded. It's usually assemblies of neurons or sort of groups of neurons that are all firing together. So the large scale firing of neuronal assemblies can be picked up by measurement devices. Sort of only large scale firing, so not a single neuron, but tens, hundreds, thousands of neurons, and there's millions of neurons in your brain. So we're still talking about sort of small spaces, but there needs to be sufficient volume of neurons acting at once and together for that activity to be picked up by our measurement devices. When we're using fMRI, what we're measuring is metabolic activity, the metabolic signature of neurons that have fired. And when we are using EEG or MEG, we're measuring electrical activity. So a nerve impulse is an electrical event, and so it can be measured electrically. And different patterns of electrical activity in the brain can be picked up. Neurons that fire together at different frequencies, or neurons do fire together at different frequencies depending on the nature of the cognitive activity. So you can sort of see here in this GIF over here, you can see how neurons don't just fire once, they usually fire a bunch of times in rapid succession. And it's kind of like a heartbeat, right? You can, sort of a frequency. It can be firing together very quickly or more slowly at some predictable rate. And just like other kinds of electromagnetic activity that we might measure in sort of physics or engineering contexts, the frequency, how fast those neurons fire corresponds to different kinds of neural activity. And we can measure this and sort of characterize it according to frequency type. So really slow frequencies, so groups of neurons that fire very slowly and only repeat every few seconds are delta waves. And that's associated with deep dreamless sleep, so non-REM sleep, or being totally unconscious. Theta waves are associated with light sleep, deep meditation, and theta waves are used in the coordination of brain regions across sort of large areas. So it's a way that brain areas from different parts of the brain communicate with each other, as well as memory recall. Alpha waves are sort of signature of a conscious state of being relaxed, awake, such as sort of light meditation or mind wandering. And this is often seen if you sort of are awake, but you're very relaxed, such as when you have your eyes closed or your mind wandering. Beta waves are associated with sort of normal alert waking state. So it's what we see when you're sort of actively concentrating on something or you're sort of actively alert. And then gamma waves are sort of the highest level, the fastest frequency level in the brain, and it's associated with higher order mental processes. So commonly associated with like learning new information, embedding new memories, having insights where when you suddenly realize something and it sort of feels like a burst, that is a gamma burst. So what we see here is this sort of spectrum from being totally unconscious to being in some extremely heightened alert state associated with very active processing, going from lower frequency to higher frequency. So this is the strength of recording the electrical activity of the brain, is that you can actually kind of understand not only that there is activity happening, but what kind of activity. What does the electrical activity tell us about what's going on? Moving to sort of the parts of the brain, so just looking at the structures at a global level, the main parts of the brain are the cerebrum, which is this big part here, which is usually what we think of when we think of the brain. The cerebellum is a structure underneath and to the back, and the brainstem, connects the cerebrum and cerebellum to your spinal cord and the rest of your nervous system in the body. When we talk about cognition, we're usually talking about processes that are happening in the cerebrum, but also, of course, involve the cerebellum and brainstem. But the cerebrum is the most sort of evolutionarily recent and advanced part of the brain, and it's where higher order cognitive processes take place, sort of the outer shell of the brain. The cortex is the surface of the cerebrum, so sort of the outside of it rather than the inside. So when we talk about the cerebrum, there's sort of the outside, which is the cortex, and inside of the cortex, there are subcortical, so below the cortex structures that sort of have specific, they're like little organelles in the brain, basically, and they have different structures and functions, and they usually correspond to some particular function. So we'll just go over each of these parts of the brain fairly briefly. So as a student of cognition, you should be familiar with what each of the lobes of the cerebral cortex do, where they are. You should be able to recognize them and to sort of have a very basic understanding of what they're associated with. So this is not like a finite list, but these are the key functions of each of the cerebral, the lobes of the cortex. So the frontal lobe is associated with executive function, concentration and attention, sort of elaboration of thought, active concentration and elaboration if you're sort of trying to think through something. It's where it helps us make judgments and decisions. So if you're sort of actively trying to reason through something, that usually involves the prefrontal cortex, which is sort of the most front part of the frontal lobe. inhibition, so like not just doing something, but deciding to not do something. It's where memory for habitual behavior is stored and motor control. And that's because the primary motor areas and supplemental motor areas are also part of the frontal lobe, over here where the cursor is. And then the prefrontal cortex is here, and that is responsible for these sort of higher order executive functions, like judgment, decision making, self-control, things like that. And this is the interior view. So if you take each of the hemispheres of the brain and you look down the middle, this is what the frontal lobe looks like. And as you can see, it takes up sort of the largest amount of real estate of the cerebral cortex in humans. The temporal lobe is this green area here. And the temporal lobe, the main functions are auditory reception and processing. So it's, as you can see, it's kind of right under where your ears are. So it's where auditory information is received and processed from the primary auditory areas. It is also a big hub for language, memory, and for just content. Like it's where a lot of your memory is stored about objects, about things, about events, categories, concepts. So what we would refer to as semantic memory. The inside the temporal lobe is on both sides are the hippocampus and the amygdala, which are sort of responsible for laying down new memories and processing memories. And then memories, once they're formed, are stored a lot of the time in the temporal lobe. So it's sort of like the filing cabinet of the brain, as well as related to auditory processing and also the high level manipulation of memory organized into categories and concepts. The parietal lobe is sort of up here in between the frontal lobe and the back of the brain. And the parietal lobe processes sensory information. So it is where the primary sensory areas of the brain are for touch and feel. It processes goal-directed movement, your sense of touch and smell, how you manipulate objects. And so this is sort of manipulating and having memory for sensory information and for a perception, right? So if you're mentally manipulating something in your mind, like trying to do mental rotation, or you're processing information about where something is in your environment spatially, that's the parietal lobe that's responsible for that. And then the occipital lobe is all the way in the back of the brain. And the main function of the occipital lobe is the visual processing. So it receives information from the eyes, and the sort of initial processing of visual information happens in the occipital cortex. And it seems relatively small relative to the other lobes, but in humans, in healthy individuals, have really good eyesight compared to most other animals. And so it takes up proportionately a lot of real estate relative to what other animals have, the amount of their brain that's devoted to visual processing. So the occipital lobe is processing information from vision, and it is helping us do initial categorization of things, objects, identifying colors, shapes, etc. And then the sort of downstream, more detailed processing of visual information usually happens in the temporal lobes and the parietal lobes. Okay, briefly, there are lots of subcortical structures in the brain, in the brainstem, etc. But for our purposes, I'm just going to point out a few that you should be familiar with and aware of. So the amygdala is this sort of green little sort of button here. It's not a button, it's a little structure. And the amygdala is responsible for processing of emotion, but specifically fear. Um, Fear and threatening stimuli, and also just sort of arousal in general. The amygdala is most commonly associated with fear, although it can process sort of intense happy emotions as well. So think of it as sort of fear and intense emotions. It's sort of this, it helps us detect and gives rise to intense feelings that are important to our survival. The hippocampus is this sort of like purple thing here. These aren't the actual colors in the brain. This is just color coded. And there's one on each side, the amygdala as well. And the hippocampus is responsible for the processing of memory, laying down the memory specifically related to like events and semantic information. So when when something happens to you, the thing that helps you encode it as a memory is the hippocampus, and then you're able to recall it later thanks to your hippocampus. The basal ganglia is largely this sort of blue thing here. There are subcomponents of the basal ganglia that we may mention, but sort of broadly, this structure is largely responsible for the processing of of rewarding stimuli. It is a huge part of dopamine pathways in the brain. So, sort of the creation and processing and transmission of dopamine throughout the rest of the brain. And it encodes memory for how valuable things are. So, if you eat the most amazing piece of cake you've ever had and you just feel all this dopamine, it's going to be sort of highly memorable because the basal ganglia has sort of enabled the encoding of that memory. So it helps us remember when things are highly valuable or when they're not highly valuable. So it's sort of, there's a sort of, dopamine is sort of the reward currency of the brain. So it's keeping track of the value of things. And it's facilitating in motivation and movement, and of course, pleasure, because pleasure is associated with rewarding stimuli. The thalamus is sort of behind or underneath the basal ganglia, and it also has a lot of different substructures, but just broadly speaking, it processes information from the senses. So we have different senses. We have vision, we have hearing, we have touch, taste, smell. All of these are sort of initially processed in different areas of the brain, and then the thalamus integrates all of that information so that you can have a cohesive sensory experience, so that you're not sort of separately experiencing what you see and what you hear and the temperature and what you feel, that it's sort of, it gives rise to really your conscious experience. When there's something wrong with the thalamus, if it's selectively lesioned, if it is malfunctioning, then this can result in sort of loss of consciousness or loss of sort of a cohesive conscious experience. So the thalamus may be sort of the closest thing that we have to that sort of central point in our brain that integrates information and gives rise to what we experience as consciousness. And then the hypothalamus, hypo means below. So this is a structure that's below thalamus. You can see it in orange there. It plays an important role in maintaining homeostasis. So it sort of regulates our body functions, including our hormones. It is part of what tells us that we're hungry, that we're tired. It helps us sort of be alert, awake when we need to be, tired when we need to be, etc. So it's it has all these sort of bodily regulatory functions. Okay, let's talk about sort of common ways that the brain is measured through neural recording and imaging methods. And sort of an important note here is that when we talk about measuring brain activity, people usually say neuroimaging as the sort of catch-all phrase, neuroimaging. But there is a difference between imaging and recording, and different techniques do different things here. So when so recording techniques involve sort of taking some direct measurement of activity from the brain, and this is usually the electrical or magnetic activity of the brain produced in a single dimension. So single cell recording, EEG or MEG recorded from the scalp, or intracranial EEG. Imaging is like taking a picture of something. So you're not recording it directly, you are sort of capturing a signature of it. So whereas EEG is like a microphone where you're actually listening to something, imaging is like taking a picture of it. And then if you look at that picture, you're not actually looking at that thing, you're looking at a representation of that thing. And that includes PET, fMRI, diffusion tensor imaging, and FNIRS. And then stimulation techniques involve producing or inhibiting neural activity through the application of magnetic or electrical currents to the brain. And that those include TMS and TDCS. And different sort of imaging and recording techniques sort of operate at different levels of spatial precision and in time. So they vary in terms of time and space. So, and then there's this third axis here of portability. So the highest spatial resolution, so with spatial resolution here on the x-axis, the highest spatial resolution, so sort of the smallest area that you can record, would be implanted EEG, where you are taking recordings of single cells or a small area, a very small area of maybe a few tens or hundreds of cells. fMRI is sort of somewhere down here, so it covers a larger spatial resolution. You can only pick up the activity of sort of many more neurons, so hundreds of neurons, and you're seeing sort of like that blob of activity that actually corresponds to sort of centimeters in the brain. PET as well, but PET and fMRI have low temporal resolution, which means that it's recording over sort of longer periods of time. And so you can't pick up very fast neural activity. EEG and MEG have very high temporal resolution, so they're picking up activity that happens very quickly, but they have slightly lower spatial resolution than PET and fMRI. And then FNIRS is functional near infrared spectroscopy, sort of functions similarly to fMRI. Okay, so here are the recording techniques, starting at the lowest level of analysis with single cell recording. So single cell recording involves taking an electrode that is incredibly small and sticking it in a single neuron, or really what you're most likely recording are sort of groups of neurons. So the pros, and then you can sort of measure the electrical activity. You can measure every time it fires. The pros of this technique are that it's obviously incredibly precise. It's really amazing if you can record at that level, but the cons are that it's very invasive. You could only do this in animals or in patients that are having surgery, their brains are already open. There's really no ethical reason for you to do this on cognitively healthy individuals, because how could you put an electrode inside of their brain? These techniques have yielded a ton of really important information about sort of the basic function of neuronal transmission in the brain. So for example, single cell recordings of the cat visual cortex have provided insight into how different neurons sort of act as different feature detectors. So there's very specific neurons that become activated in response to a very specific kind of stimuli. It's like a horizontal line versus a vertical line. And you can see actually here kind of more what it looks like. So if this is an electrode and these are neurons, you're recording the activity of on the order of magnitude of like tens of neurons, right? You're not sticking neurons are too small for us to be able to have little tiny pieces of metal that could actually hit the cell body. So in humans or other mammals, at least, that's sort of what you're recording. Also, recordings of sort of primate dopamine neurons have demonstrated the neural basis for operant conditioning. The fact that neurons will sort of fire first in response to a reward and then in anticipation of a reward from a cue. One sort of level of analysis bigger is electrocorticography and intracranial EEG. And this measures electrical activity directly from the cortex or within the cortex using electrodes. So what you see here on this picture is kind of like a mesh where the literal physical cortex has an electrode laying on top of it. So you're measuring activity directly from the brain. So you're measuring it by touching it. And then depth electrodes are implanted into the electrode or into the cortex, and you're measuring at different points in vertical space from an electrode that has sensors on it that are all lined up together. So it enables a sort of detailed mapping and precise measurement of electrical activity, like directly from the actual organ. Again, the pros are that this is incredibly spatially precise and there's very high temporal resolution, but it's very invasive and it can only be done on animals or surgical patients. This is done in patients who have epilepsy, and they will need to undergo brain surgery to get the part of their brain removed that's causing their seizures. And in order to figure out the part of the brain that's causing seizures and the part of the brain that's not causing seizures, you need to record lots of activity from all these parts of the brain directly so that you can localize that part and only take out exactly what is needed. And then through this, we're able to learn a lot about how different parts of the brain function, but you can only do it in this very limited clinical population. What we would do with a healthy adult or someone where their brain already isn't exposed, their skull isn't cracked open, is EEG, which stands for electroencephalography, or MEG, which is magnetoencephalography. And here you are measuring the electrical activity of the brain, but from the scalp, from outside of the head. So whereas the intracranial recordings or the electrodes are directly on the brain, here the electrodes are outside of the brain over the skull. So you're getting sort of being able to record directly from the brain is pretty amazing, but usually we don't have that luxury. Here we're measuring the electrical activity through the skull and the other tissue between sort of the outside of their head and the brain. EEG has really great temporal resolution, meaning that you're picking up really fast neural activity, and picking up activity in those different frequency bands tells us a lot about the kind of activity that's happening and where it's happening from. So the pros are that it has high temporal resolution, it picks up the whole spectrum of electrical frequencies, it's easier, it's more portable and less expensive than fMRI. When you're able to record EEG at these really high densities, you can sort of do these reverse inferences into where the activity actually happened from. It's also a lot less expensive than fMRI. So that it's often used for that. The cons are that it has a lower relative spatial resolution than fMRI, and you're only picking up activity from the very sort of outer parts or the cortex or the top of the brain. It can't really pick up any electrical activity that originates any deeper than the surface of the brain. And here's how this is what EEG data would look like from recording. That can be either averaged into these waveforms that we call ERPs, which are called event-related potentials. And we can sort of look at the different form and structure of these ERPs in response to different stimuli. You can also make inferences about where electrical activity happened if you have enough density at the cortex. You can sort of do this reverse inferential process that enables you to sort of generally be able to isolate where activity originated from, even using EEG. Okay, now let's talk about imaging techniques. So PET stands for positron emission tomography. This was the earliest form of imaging, and it's not really used today because it involves ingesting a small amount of a radioactive substance that can be detected in the blood. So what you're really doing when you're measuring neural activity using a PET scan is you're measuring blood flow, because you're picking up the radioactive signature of this particular substance. And here, blood flow is assumed to reflect greater neural activity, so more metabolic activity, more neural activity. So if you see more of this radioactive substance in a particular area when someone is doing a task, we infer that that area is related to the processing of that task. fMRI has really made this obsolete because in fMRI, you don't have to have people ingest a radioactive substance to basically get the same effect. So let's talk about fMRI. fMRI is an imaging technique that indirectly estimates neural activity by measuring changes in blood oxygenation within the brain. So it's measuring how much oxygen is in the blood in different parts of the brain. And the more oxidized, or the greater the sort of amount of oxygenated blood in an area, we assume reflects metabolic activity. So that part of the brain is more active, so it was doing more. This sort of gives rise to a three-dimensional image of the brain's metabolic activity over some period of time. And the units of this sort of three-dimensional image are called voxels. So a pixel is a two-dimensional sort of unit in an image, and a voxel is a three-dimensional unit. So these little cubes. Machines with stronger magnets can generate images with higher spatial resolution, so more precise. So stronger magnet equals greater spatial precision. And here, like I said, areas of the brain that receive relatively more oxygenated blood are assumed to be more active during a particular task. So the pros are that it's better able to isolate the spatial activity of a of activity in the brain, but it takes place over a longer period of time because blood flow happens sort of slower than electrical activity. But another pro is that it can pick up metabolic activity throughout the entire brain, including the subcortical areas, whereas EEG really can't do that. It is quite expensive because running an MRI machine is very expensive. It requires people with a lot of specialized knowledge, and it's not invasive, but it's kind of uncomfortable. And if anyone has ever had an MRI for anything and you are sitting there in that spinning donut, it's really, really loud, and it kind of is like there's like a banging sound. So if you think about trying to do a cognitive task, like thinking, remembering, solving a problem while you're in this giant, loud, thumping donut, it's kind of uncomfortable and it's kind of distracting. So it is just useful to point out how, although this is a really good way of measuring neural activity, it doesn't exactly it's not exactly a conducive environment for a lot of cognitive processes. So there's a trade-off there. What you can get from fMRI are images that look like this, where we can see relatively greater areas, areas that showed greater neural activity versus less neural activity during a particular task. From fMRI information, we can also sort of generate images of the structure of the brain, right? Because we can look at these sort of fine level of detail of how blood flows, so we can really map tracks of neurons. FNIRS stands for Functional Near Infrared Spectroscopy, and the way that it works is that it emits infrared light into the brain through the skull because it's able to permeate the skull, and then it measures its refraction. So it's kind of like echolocation, but using light. So it transmits light into the brain, and then it picks back up what is reflected back, and that enables you to sort of generate a picture, an image of density of tissue at different levels, right? Just like echolocation. So tissues of different densities will refract at different wavelengths, which allows for reconstruction of neural activity, because brain tissue that has like greater blood flow, greater oxygenation level, will refract back at a different wavelength than brain tissue that has lower levels of blood flow or metabolic activity. So it kind of is measuring something similar to what fMRI is measuring, but using a different technique, using light refraction rather than measuring the radioactive signature of blood oxygenation. The pros are that it's way less expensive than fMRI. It kind of has the same physical structure as EEG. It's a cap. So it's it's easier to deploy and use, similar to that of EEG, and it's less invasive. It's not loud. It doesn't have that big thumping, loud, noisy fMRI. And it's often portable, so you can potentially use it in like real life situations. It is good. One of the cons is that similar to EEG, you're only really able to measure activity from the very top of the cortex. It doesn't permeate very deeply. And so it has trade-offs relative to fMRI, but it's good if you are interested in questions that fMRI could answer. But for populations that fMRI is not applicable to or not good for. So like very small children, people who are elderly, people who like would feel physically uncomfortable or stressed out to be in an fMRI machine, or people who are neurocognitively impaired and couldn't would have a difficult time sitting still and being calm in an fMRI. It's also very portable, right? So you can bring it to different places. You could bring it into a school. You could bring it into a place of business. You can have someone wear it while they're doing something in a semi-natural way. But it has the same drawbacks of EEG, that it's only able to measure the small amount of the cortex, and it has the drawback of fMRI that it is measuring activity over a lower temporal resolution, so only over the course of seconds rather than milliseconds, is what you go with EEG. Okay, last techniques are stimulation techniques, and rather than picking up the activity from the brain, what you're doing is stimulating the brain and measuring people's behavior as a result. So you're not just measuring activity, you're actually changing neural activity through electrical stimulation or magnetic stimulation. So you can jolt someone's brain with a current of electricity or a magnetic pulse, and this causes and stimulates neuronal activity in the area under which you stimulated. Then you measure the way that affects their behavior, and that helps you causally be able to say, okay, we stimulated someone's area of their prefrontal cortex, and then they were better able to pay attention in this task than the control group that did not receive stimulation. Therefore, we know that this area must be associated with enabling attention or concentration, for example. So you're really able to do some very important scientific experiments because you're able to sort of directly say that this causes this. Um, Also, stimulating different parts of the brain seems to be really clinically helpful in certain situations where there may be some sort of dysregulation of a brain area, a brain region, or sort of how it is associated with some process. And stimulation can kind of jumpstart it. It's kind of jumpstarting a car and sort of refresh the batteries and enable better regulation. This is particularly seems to be the case for depression. So TMS and TDCS are now treatment methods for depression that's resistant to like pharmacological interventions. So like taking an SSRI, that seems to really help people. The mechanisms are that, or the cons are that we don't really know how it works. We just know that it does work and that the spatial precision isn't great, especially for electrical stimulation. So you're kind of just spamming a kind of large part of their brain with electrical activity. And so we can't make very fine-grained, precise conclusions from that just because we're limited by sort of, you have to be hitting their brain and the signal is fairly big. So you're hitting a large area. Some other just last measures here are not neurological, but they reflect neural processes. So other kinds of physiological things that you can measure include electrodermal activity, so skin conductance. How much the conductance of someone's skin changes is a signature of their the sympathetic nervous response to stress and arousal. You can do ECG or EKG, measuring heart rate, and you can measure people's pupil dilation. So pupils dilate not only in response to, or contract not only in response to sort of like the level of ambient light, but also pupil dilation reflects arousal, stress responses, and it's a proxy for the release of dopamine, adrenaline, and serotonin. So you can literally see, for example, when people have a creative idea, have an insight, their pupils dilate, right? Because it reflects that dopamine has been released, or adrenaline, for example. So these are other parts, bodily signatures that you can measure to make inferences about neural activity. All right, I'm going to stop there. I know that was a lot of information, but all this information is also available in the textbook and in chapter 2. And on Wednesday of this week, we will talk a little bit more about why and how we measure the brain in relation to cognition and some sort of philosophical debate about what we're trying to achieve when we do so. Great. Have a great day.